12th Bio-botany, Chapter 4, Principles and Process of Biotechnology. In this part, today we shall discuss about vector-mediated gene transfer. It is an indirect method of gene transfer. In the previous part, we discussed about direct method of gene transfer. Vectors are used to carry foreign gene into plant cells. Foreign gene is called as gene of interest. This is a vector, name is PBR322. This is a specially designed vector used for the purpose of gene transfer. Most commonly used vector is TI plasmid. The TI plasmid has virulence region. It is a disease causing DNA sequence. This DNA sequence causes disease in plants, a cancer like disease. And this TI plasmid also has tDNA region. tDNA region is called as transfer DNA. This causes transfer of gene into plant cells. This TI plasmid is present in Agrobacterium tumefaciens. Agrobacterium tumefaciens is a bacterium. It has TI plasmid. It is a disease causing bacterium. It causes crown gall disease in plants, a cancer like growth. This Agrobacterium tumefaciens bacterium has a large size plasmid known as TI plasmid. It is a tumor inducing plasmid. The TI plasmid has a part called as tDNA. A portion of TI plasmid is referred as tDNA, transfer DNA. This transfer DNA involves in the transfer of genes into plant cells. The bacterium transfers transfer DNA to plant genome in the infected cells and cause plant tumor called as crown gall disease. The infected cells develop tumors. The bacterium causes infection through the damaged area of the plant. The tDNA region of plasmid is transferred into plant genome while causing infection that results in disease. This bacterium infects cells at the wound site and transfers tDNA region of its plasmid into the plant genome. Naturally, this bacterium transfers tDNA region into plant genome. So this bacterium is called as natural genetic engineer of plants. We have already learned that the TI plasmid has virulent region, a disease causing region that causes disease. Then how is TI plasmid used? Let us see. The unwanted sequence in TI plasmid is removed, and in place of unwanted DNA sequences, needed sequences are added. Disease causing unwanted sequence in TI plasmid is removed. In that place, needed sequences are added. After adding the needed sequences, it is called as recombinant TI plasmid. Now let us see the needed sequences. The first one is gene of interest, or we can say foreign gene. The foreign gene is inserted in the place of unwanted. The unwanted sequence is removed. In that place, needed sequences are added. This is needed sequence called as BT gene. BT gene is isolated from a bacterium called as Bacillus thuringiensis. The Bt gene produces a toxic protein that kills caterpillar of insects. In that way, the Bt gene provides resistance, insect resistance. This foreign gene or gene of interest is inserted. Then plant selection marker gene, usually an antibiotic gene like NPT2 is inserted. The antibiotic gene provides resistance to antibiotic kanamycin. The uses of marker gene we will discuss in the next video. So after the insertion of needed sequences, it is called as recombinant Ti plasmid. Agrobacterium, this is Agrobacterium, this one is genomic DNA, this is plasmid. The plasmid is taken out, it is opened by restriction enzymes, cut by restriction enzyme to remove the unwanted sequences. In that place, the foreign gene or gene of interest is inserted. The gene of interest is joined with the Ti plasmid with the help of the paste molecule DNA ligase. DNA ligase joins two DNA molecules. As a result of this, rDNA is formed. This is recombinant Ti plasmid. This recombinant Ti plasmid is transferred into plant cells. So we have to transfer the recombinant Ti plasmid into the plant cell. Before that, the recombinant Ti plasmid is inserted into the Agrobacterium. The Agrobacterium is allowed to infect the plant cell. While the Agrobacterium is allowed to infect the plant cell. At that time, the transfer DNA, tDNA, causes transfer of the foreign gene into the plant genome. Here the plant genome is given in green color. What's the color of Ti, the foreign gene? Red color. While infecting the plant cell, the tDNA causes transfer of the foreign gene into the plant genome. The plant cells are now called as recombinant cells. The cells containing foreign gene. The cells are cultured or incubated in a plant tissue culture medium that results in the development of plant with foreign gene. This plant is genetically modified plant. The plant has new trait, new characters. In this way, Agrobacterium is used to mediate gene transfer in plants. In short, we can say the unwanted part of the Ti plasmid is removed. In that place, needed sequence of DNA is inserted. The bacterium is allowed to infect the plant cells. At that time, it transfers the foreign gene into the plant genome. Then the plant cells are cultured to produce genetically modified plant cells. Screening a recombinant is important in recombinant DNA experiments. After the experiments, we have to separate recombinant cells from non-recombinant cells. Recombinant cells have the inserted foreign DNA. Non-recombinants don't have. The separation of recombinant cells from non-recombinants is called screening. After the introduction of rDNA into a suitable host cell, it is essential to identify those cells which have received the rDNA molecule. This process is called screening. Recombinants have the foreign gene or inserted gene. The vector or foreign DNA present in recombinant cells expresses the character The recombinant cells have foreign DNA, so the character of foreign DNA is expressed in the recombinant cells, but non-recombinants, while the non-recombinants do not express the characters or traits. We shall discuss about the screening process in detail in the next video.